Okay, thank you. Thanks for the very nice introduction, and I would like to thank the Neurological Foundation for the opportunity to share some of my research here today. So today I'm going to talk about auditory condition called tinnitus. And so at the beginning, what is tinnitus? Tinnitus is a ring, buzzing, or roaring sound in a person's ear or a person's head without the corresponding external sound. For most of us, tinnitus could be very transient. It may last for a few seconds or a few minutes, then it will go away. However, for people suffering from chronic tinnitus, it is a very debilitating condition. As described by Pavlovian for his tinnitus, he said, "My ears whistle and buzz continually, day and night. Such a condition is truly frightful." So you can feel the frustration there. So tinnitus is actually a fairly common condition, probably more common than people normally think. So the prevalence of tinnitus is pretty much very similar across the world. So it is about 10 to 15% in the adult population. However, tinnitus prevalence increase with age. As you can see in people over 60 years old, the tinnitus prevalence can reach to about 25%. It has been estimated that in New Zealand, about 200,000 people are affected by tinnitus. Also, tinnitus prevalence can get quite high in certain occupations where people are exposed to loud noise, such as in the construction industry, the manufacturing, and in the music industry, and also especially in the military population. So in those occupations, the prevalence of tinnitus can get up to 30%. Very interestingly, recent years, there is people have noticed that there is an increase in tinnitus incidence in younger population. This is because young people in these days spend prolonged time listening to loud music through headphones, and also going to a nightclub, going to concert where the music played very loudly. So there has been some study to look at the link between COVID-19 and tinnitus and other auditory problems. Also, it's in a very early stage. There is a study conducted in Italy in 2020 where they looked at 185 COVID-19 patients, and they found that about 23% of them experienced tinnitus. It is still too early to know whether tinnitus in those COVID patients is transient or they will last for a long time. So it's too early to know that. And also there are some studies looking at the the side effect of the COVID-19 vaccination. And they found that there are very small percentage of people who received COVID-19 vaccination. They also experience some new forms of autological problems, or the vaccination made their auditory problems worse. So this includes tinnitus as well. But again, it's still in a very early stage. And as we know, there are lots of research into develop the treatment for COVID-19. So there are some promising treatment which can reduce the hospitalization for the COVID-19 patients and also reduce death rate. However, it has been noticed that some of those treatment may, some of these drugs may have the potential to induce tinnitus or other auditory problems. There are lots of factors can cause tinnitus. About 90% of people with tinnitus or experience some degrees of hearing loss, whether it is noise-induced or age-related. However, tinnitus can occur without detectable hearing loss, for example, after head or neck injury or in Meniere's disease. Tinnitus can also occur when there is a tumor grow on the auditory nerve. In addition, there are about over 400 medicines out there that can either trigger tinnitus or make tinnitus worse. So among all these factors, noise exposure is the most common cause of tinnitus in humans, which is not surprising. So as I mentioned before, tinnitus can be very debilitating, and about 30% of people with tinnitus found that their tinnitus is annoying, but it doesn't really affect their normal life. However, most of them, about 60% of them, tinnitus causes various problems in their life. For example, people may have difficulties in sleep, they may have trouble in concentrating, and there we have seen high incidence of anxiety and depression in tinnitus patients. And also some patients feel like they are isolated because of their condition. And there are about 2% of people who are really affected by tinnitus and they cannot carry on with their normal jobs. So in severe cases, tinnitus can even lead to suicide, which is very sad. So it would be ideal if we can have a treatment that can simply turn off the tinnitus sound. However, none of the currently available tinnitus treatment is able to achieve that. There are some tinnitus management currently available. For example, the acoustic therapy, where people can listen to other types of sound or music to mask the sound of tinnitus. So tinnitus is not that obvious to them. Alternatively, cognitive behavior therapy or psychological counseling or relaxation training, these can help people to train them to shift their attention away from tinnitus. So tinnitus is not that annoying to people. So all this management focus on reduce tinnitus-related distress, but none of them can make tinnitus go away. More recently, transcranial magnetic or electrical stimulation has been trialed in tinnitus patients. However, there is no evidence for their effectiveness in reduce the loudness of tinnitus itself. And when we look at the medicines for tinnitus, this is very unfortunate. There is no a single drug that has been approved for the treatment of tinnitus. So in summary, currently there is no effective treatment for tinnitus. So this is mainly because we still do not fully understand how tinnitus is generated. So in the earlier days, it was thought that tinnitus is generated in the ear. This is because there is a strong association between hearing loss and tinnitus, and also in animals after noise exposure, we can see hair cells, which is the tiny cell in the ear, which is responsible to detect the sound. So these hair cells, you can see they get damaged after noise exposure. And also we see the damage of the synapses, which is the connection point between the hair cells and the auditory nerve. They get damaged too. So it seems logical that tinnitus should be generated at the place where the damage is. So if this is the case, we should be able to treat tinnitus by removing the pathological source of tinnitus. So the doctors have tried to do that. So what they did is they just they did the surgery to cut the auditory nerve. So the idea was that if the tinnitus is generated by those damage in the ear, then by cutting the auditory nerve, the tinnitus sound cannot be heard by people. However, this procedure could not eliminate the tinnitus sound. In some cases, it even made tinnitus worse. So this suggests that the damage to the inner ear may be just a trigger of tinnitus. The generation and the maintenance of tinnitus may occur at a higher level. So there are lots of evidence to support this idea. For example, in animals, people are able to record neuronal activity in different areas along the auditory pathway. As we know, when we hear a sound, the information of the sound is transmitted along the auditory pathway, firstly to the lower part of the brain. There is a area called cochlear nucleus, and then it moved upwards to inferior colliculus and thalamus, then finally reach the auditory cortex, where the sound is perceived, can be heard by us. So what people found in animals, after noise exposure, neurons in different parts of the auditory pathway become hyperactive. So neurons send out activity more often, and also more neurons fire at the same time. So it's over active. And so it has been suggested that tinnitus may be generated by those hyperactive neurons in the auditory pathway. And then with the advanced imaging technique, people are able to view the neuronal activity in different parts of the human patients. So, what people have found is that in addition to the activation of the auditory pathways, there are activation of many other non-auditory neuronal networks. So these just show you the activation of the cognitive control network here. And also you can see the activation of salience network. So put all the findings together, we can see that in tinnitus patients, we can see the activation of not only the auditory networks, which I have explained before, but also activation of the distress network. So these areas are responsible for the emotion control. So the activation of the distress network may be responsible for the people's reaction to tinnitus. So responsible for tinnitus-related distress. And we also see the activation of the memory network. These include the structures such as hippocampus and parahippocampal areas. So these areas play a very important role in different types of memory, including the auditory memory. So it has been suggested that tinnitus signal may be memorized in these areas, so tinnitus become persistent in people. And we also see the activation of the conscious perception network. We know that the sound generated from the auditory pathway, they cannot be perceived or heard by us unless the auditory pathway is connected with the perception network. So we see the activation of the conscious perception network and the increased connectivity between the auditory network and the perception network. This suggests that these changes may strengthen the perception of tinnitus. So the tinnitus sound become very obvious to people. So as I said, tinnitus, as you can see, tinnitus is not simply a disorder of the ear, but a disorder of the brain. So this involves multiple neuronal networks, not only auditory network, but a number of non-auditory networks. And also the changes is not only in the activity of those networks, but also the connectivity between different networks. So it's very complicated. So we think that the most effective tinnitus treatment should target at those tinnitus causing networks rather than the single molecules or single receptor or single brain area. So in our lab, we are doing tinnitus research using an animal model of tinnitus. We aim to understand the mechanisms of tinnitus by looking at the neurochemical basis of tinnitus and also the electrophysiological mechanisms of tinnitus. We also try to identify biomarkers of tinnitus, and we also evaluate some potential treatment for tinnitus. So talking about animal model of tinnitus, we have this rat model of tinnitus, which is induced by noise exposure. So as I said before, noise exposure is the most common cause of tinnitus in humans. So this animal model is regarded as the most realistic animal model to represent tinnitus in humans. And also we found, and also other people in other parts of the world, they use the same model. They found that noise can cause tinnitus in animals that last for a long period. So this can mimic the chronic nature of tinnitus in humans. So we have the animal model we have here is the only animal model of tinnitus in New Zealand. So every time when I talk about my tinnitus research using animal model, the first question I always get from people is, how do you know a rat is having tinnitus? This is a very good question because tinnitus is subjective and we cannot hear the tinnitus sound in animals' ear or in their head. The rat obviously cannot come to us saying, hello, I'm having rain in my ears. So we have to develop an objective measure for the perception of tinnitus in those rats. So we develop a behavioral paradigm, which is based on the Pavlovian conditioned response, where a conditioned stimulus, when it paired with an unconditioned stimulus over a period pairing, the animal will develop a conditioned response in response to the conditioned stimulus. And then over a period, even without the presence of the unconditioned stimulus, the animal will still respond to the conditioned stimulus. So in our behavioral paradigm, we use silence as our conditioned stimulus and mild food stimulus as the unconditioned stimulus. Then we use the separation of the drinking activity as the conditioned response. I will just explain it a little bit more in details. As you would imagine, this is a very complicated behavior paradigm, but I will try my best to explain it in a very simple way. So hopefully it's a bit easy to understand. So basically in normal animals, when we pair the silence with food stimulus, they will produce the separation of drinking activity, which is measured by the number of licks the animal doing. So we will see licks separation in those animals in response to the silence. And then we give them a number of testing stimulus, which is sound of different frequencies and intensities. And as we know, those sound is very similar, very different from the silence. Okay, so when the normal animal hear those sound, they will not have lick separation behavior. However, in tinnitus animals, they will hear their tinnitus during the silence period. Okay, so it is their tinnitus instead of silence paired with the food stimulus that caused lick separation behavior in them. So then when we play sound of different frequency and intensities, if some of these sounds very similar to their tinnitus, the animals will also respond to this sound to have the lick separation behavior. So that's, can you see the difference between tinnitus animals and normal animals? Their different behavior to the different sound we played to them. So that's how we can tell whether an animal is having tinnitus or not. So when we look at our results, we express them as frequency discrimination curves. So as you can see here, when we play the broadband noise to the animals, the sham, which is the normal animal, and exposed is the noise exposed animals. So their behavior are quite similar. So there is no difference between these two lines. When we play the 10 kilohertz sound to the animals, their behavior is still pretty much the same. However, when we play the 20 kilohertz sound to the animals, as you can see, the noise exposed animals has responded to this 20 kilohertz sound and suppressed their drinking activity. So which suggests that the animals may experience tinnitus, which sound very similar to the 20 kilohertz sound. So use this animal model, we have done various experiments to look at, to investigate the mechanisms of tinnitus, to evaluate drug treatment. Here, I just want to focus on one particular study where we use the animal model to look at tinnitus causing networks using a novel technique called metabolomics. So metabolomics is a large-scale study of the small molecular metabolites and metabolic pathways within cells, tissues, or organisms. Why we are interested in metabolites? As you can see, metabolites are sitting in a very unique position here, where they receive all the information from the upstream omics and also the information from the external environment. So this makes the changes in metabolites very sensitive, reliable, and powerful in reflecting changes in relation to either disease or drug action. So metabolomics actually has been applied very widely for diagnosis and monitoring disease progression, and for monitoring drug effects, and for develop personalized medicine. And it has been applied to many conditions such as cancer, diabetes, cardiovascular disease, Alzheimer's disease, and so on. However, it has never been applied to tinnitus research. So we conduct this world first investigation using metabolomics. So firstly, we did this in our animals. So in our animal model, we take serum samples and brain samples from our animals, and then we measure the metabolites using this very special GCMS technique, and we can identify over 100 compounds in those samples, and then use some specialized mathematical method. We can summarize all the 100 metabolite information into this one dot. So this called the metabolic profile. So each dot represent the overall metabolic profile in one rat, and the distance between each dot represent the similarity of the metabolic profile. So as you can see here, the dots from the sham animals, which is our control animal, they are all clustered together, and dots from the exposed animal, they are all clustered together, and there is a clear separation between the control animal and the noise exposed animals. And we can see the changes, the same pattern of separation in along the auditory pathways. So this is very exciting finding. This is the first time we show that using metabolic profile, we can detect which rat is tinnitus, which rat is normal. So this is very exciting. What is more exciting is that we also found that the metabolic profile are significantly shifted in tinnitus animals in hippocampus. If you remember, hippocampus is the area responsible for memory. So as you can see here, in different subregions of hippocampus, we see the separation of the metabolic profiles between sham and exposed animals. So in humans, in tinnitus patients, the image studies have shown that hippocampus undergo activity changes in tinnitus patients. So here we are very exciting to see that using metabolomics, we can also pick up changes in our animal model. What's even more exciting is that we see metabolic profile shift in serum samples from our animals. Why this is more exciting? Because if we want to eventually translate our findings into human use, into clinical use, and to develop these as biomarkers for tinnitus, we cannot take brain samples from tinnitus patients, of course. So we need to have sample that is easy access. So if our metabolic profile in serum can reflect what happened in the brain, this is a better way to be used as biomarker, right? So the next thing we are doing is we want to validate our these metabolic profiles to see whether they can be used as biomarkers. So for doing this, we use a drug, a traditional Chinese medicine, which has been used for a long time in China for the treatment of deafness, but it has not been tested for tinnitus. So we decide to use this drug to treat our tinnitus animals. And the idea is that if the drug can treat tinnitus in our animal models, at the same time, the drug can normalize the shifted metabolic profiles in those animals, it would prove that these metabolic profile changes can be can be used as biomarkers for tinnitus as well as drug treatment. Okay, so as you can see here, when we use this drug to treat our animals, the two green lines are the normal animals. So they received either the vehicle treatment or high dose of this drug. As you can see, the drug didn't make very much difference in those animals. And this red line is the exposed, noise-exposed animals that receive vehicle treatment. So as you can see, they still have tinnitus. However, the animals that receive lower dose and high dose of the drug, you can see their lines are sitting with the normal animals. So which suggests that this drug has potentially reduced tinnitus in our animal model. So we are still in the process of measuring the metabolic profiles in those animals. So hopefully we will be able to see that the metabolic profile for these animals are shifted towards the normal animal. So based on our animal results, we think it's very encouraging. So we did a pilot study in tinnitus patients using the metabolomics technique, where we have 30 controls and 30 tinnitus patients. And so we did a 4-scale clinical assessment for those tinnitus patients, for their hearing levels, for the tinnitus characteristics, and also measured the tinnitus-related distress in them, and also the mood changes, the anxiety scores and depression scores, and so on. So then we take serum samples from those participants, and then we measured the metabolic profile changes in those samples. So what we have found, firstly, in those samples, we are able to find 137 compounds. Of those, we can identify 94 metabolites in those samples. And then using the same method, we can see the separation between the metabolic profiles between the control subjects and tinnitus patients. And then we did another analysis which specifically tell us how good is the separation. And we found that there is a significant separation between the control group and the tinnitus patients. So this is very encouraging pilot study, which suggests that we can potentially using this in tinnitus patients to tell whether they have tinnitus or not. And then the next step, we want to correlate those metabolic profiles with the clinical characteristics of tinnitus. So hopefully we can subtype the tinnitus patients based on their different metabolic profiles. And then hopefully based on that, we can develop personalized treatment to target the affected network. So just talking about the development of personalized treatment. So our goal is to develop data-driven tinnitus subtyping and personalized treatment for tinnitus. So as we know, tinnitus is highly heterogenic condition. So that's why the current tinnitus treatment is not that effective, because different tinnitus patients have different response to different treatment. So hopefully using our metabolic profiles, we can subtype tinnitus patients, and then based on their different subtypes of metabolic profiles, hopefully we can develop a drug treatment that is specifically targeted to certain metabolic profiles, and then to have personalized treatment. And this drug treatment can also be combined with other forms of treatment, and based on their effectiveness on shifting the metabolic profiles, maybe we can have more effective treatment for tinnitus. So finally, I just want to thank everyone in our research group for their hard work over the years, and also my collaborators, and also the funding body for their continuous support for our research. And thank you very much for your attention. And I'm happy to take some questions if you have some. Thank you.